Hey guys, and welcome back to Now I Know. Today we are talking about translation in prokaryotes. Now, in Central Dogma, we have talked about DNA replication in prokaryotes and eukaryotes. We have talked about transcription, that is synthesis of mRNA in prokaryotes and eukaryotes, and today we will start with the last part of Central Dogma of molecular biology, that is the translation. So today we will talk about translation in prokaryotes. Now you might know what exactly is translation. It's a protein synthesis, but definition wise, when the mRNA is made, this mRNA has all the codons, all the information that is needed for synthesis of amino acid sequence or protein synthesis. So when this mRNA is converted into the amino acid sequence of polypeptides, when all these amino acids are arranged one after the another and protein is made, is the process of translation. So mRNA converted into amino acid sequence of polypeptide is the synthesis of translation. Now when it comes to translation, the molecules or the machinery that we need is mRNA, of course, because that is the information template, or that has all the codons. We have the transfer RNAs that brings in the amino acids. We have the amino acids itself, ribosomes where the translation occurs, and other accessory proteins that are needed for synthesis of protein. We will just have a look briefly on mRNA, tRNA and ribosome, because I think that will help us a lot to understand the actual process. And trust me, the whole machinery or the whole mechanism of translation is one of my favorite in Central Dogma. It's amazing. It's really easy to understand. I have shown most of the diagrams here so that it's easy for you to visualize and understand. So let's begin with mRNA first. Now when it comes to mRNA, we know it's a molecule that has all the information for the protein synthesis. But this mRNA has three parts. Okay, 5' untranslated region, there is this 3' untranslated sequence, and a reading frame. Okay, so a reading frame is easy to understand, is actually the portion of mRNA that has all the codons. Alright, and all the sequences will be there, and codons, we know they are in triplets, right? So there will be all the codons that codes for three nucleotides together form one amino acid. So this is the reading frame that contains a start codon, the sequence of amino acid, and a stop codons. Now the start codon is always AUG, that codes for methionine. Alright, always it is the AUG that codes for methionine, and it ends with stop codons, and there are only 3 stop codons that do not code for any amino acid. So when these codons come, it is the termination of translation. So these codons are UAA, UGA and UAG. Any one of these will be there. 5' untranslated leader sequence, that is the upstream or before the start codon. Simply that is also called leader sequence of 5' UTR. And then there is a 3' untranslated trailer sequence, that is after the stop codon, that is simply called the trailer sequence or 3' UTR. So there are 3 main parts of mRNA: 5' UTR, a reading frame that has a start codon, sequence and stop codon, followed by 3' UTR. Then comes the tRNA or transfer RNA. We know that it brings the amino acid to add in the growing polypeptide chain. So what happens is this mRNA has all the codons, right? All 3 triplet nucleotide making one codon for amino acid. So transfer RNA, the tRNA that brings in the amino acid, should have the counterpart or the complementary sequence. That means tRNA has the anticodon on it. Right, then only it can fit on the mRNA. So if this is the tRNA for methionine, if this is the methionine amino acid, the codon for methionine is AUG, right? So the anticodon will be UAC. So tRNA will bring in the amino acid, and amino acid is attached to the 3' end of the tRNA. That will have the anticodon that can fit on the codon of mRNA. This was just an example. So that's all in simple words about tRNA. Now about the ribosome. Ribosome is where the protein synthesis will occur, where the amino acids are assembled into polypeptide. So here the ribosome consists of two parts, a large subunit and a small subunit. The small subunit is the 30S ribosomal subunit and large subunit is the 50S ribosomal subunit. They come together, form the 70S ribosome, and that's where the protein synthesis occurs. So when this makes the 70S active ribosome, there are 3 tRNA binding site present on this ribosome. Okay, I'm saying 3 tRNA site because that's where tRNA will come and bind. So there is this one site called the E site or the exit site. So the term itself says exit means that tRNA will be released from that. P site is the peptidyl site, that means that is where the peptide bond formation occurs, and the A or the amino acyl site, that means that is where the new amino acid will enter. So this will become clear once again when we are moving on with the procedure. But remember, when the active ribosome is ready, 70S ribosome, there are 3 sites for tRNA binding, that is the E site, P site and A site, and this is how is the sequence EPA. So now let's begin, with this information, with this introduction. Let's begin with the translation. Like replication and transcription, translation also has 3 phases, and that is the initiation. The initiation has to occur, the assembly has to occur, then there is elongation, that is the formation of polypeptide chain, and of course there is the termination where the synthesis needs to be stopped. So first we'll see what is or how the initiation occurs. So initiation itself has 3 steps. Alright, we have the mRNA ready here, 5' to 3', which has the 5' UTR region, there is a 3' UTR region, there is this reading frame. You can see the start codon over here. Now, as I said, the region before the start codon or the reading frame is the 5' UTR region. In this 5' UTR region is where the ribosome binding site is residing. Okay, and that is called the Shine-Dalgarno sequence, that is simply named after their discoverers. So this Shine-Dalgarno sequence or ribosome binding site is in this 5' UTR region, because ribosome needs some indication where it has to bind. Where is the start codon? That is just before the start codon, the ribosome binding site, where first the 30th subunit will come and bind. And this binding occurs with the help of initiation factor. So that is the first step. The 30S subunit binds with the initiation factors and a molecule of GTP. So initiation factor 1, 2 and 3 along with GTP will bind to 30S subunit. Now once this binding occurs, there will be also this initiator tRNA coming into picture. Now what is this initiator tRNA? As I said, the first codon is AUG, and here it specifies the modified form of methionine, that is the formyl methionine. So you will see this F-met written in the book. So it brings in the F-met on the tRNA. So now we have the initiator tRNA and we have this 30S subunit along with the initiation factor and GTP. Both will come and bind to this mRNA. Now where it will bind? This is the second step. The initiator tRNA will bind at the start codon, and the 30th subunit can recognize the Shine-Dalgarno sequence, the ribosome binding site, that is where it will. Now this Shine-Dalgarno sequence is recognized by this 30th subunit, and that that binding site for that Shine-Dalgarno resides in the ribosome in the 16S rRNA part. Okay, so that means Shine-Dalgarno sequence is complementary to the 3' end of the 16 srRNA of the 30S ribosomal subunit. Okay, just a little more information that we should know about. So the 30S ribosomal subunit can bind properly to the mRNA, and the initiator tRNA containing F-met will bind to the start codon. So that is the second step. The first step was formation of or binding of this initiation factor with 30S and F-mat to tRNA, and the second step is once both of these are ready, they will come and bind or sit on the mRNA. 30s subunit can sit properly by recognizing the binding site, and this tRNA, initiator tRNA, has the anticodon for the start codon, so it will sit properly. Now this assembly where you have the 30s subunit and initiator tRNA bound to mRNA is called the 30S initiation complex. The third and last step in case of initiation is, now this assembly has occurred, right? What is missing? It is the 50s subunit, because we need the active 70S ribosome. So now the 50S subunit will be added to this complex and GTP hydrolysis will occur. So 50S subunit will bind over here, and this becomes now 70S subunit. So this assembly over here where you have mRNA, initiator tRNA, and 30S and 50S, that means the 70S ribosome bound to it, is called 70S initiation complex. Now this is ready to start the synthesis. This is what the initiation is. Simple to understand. You have the 30S coming into picture with initiation factor 1, 2, 3 and a molecule of GTP. Initiator tRNA bringing in the modified form of methionine, that is the formyl methionine. 30S and tRNA binds to mRNA first, making the 30S initiation complex. What is missing? 50S subunit, that will be added, GTP is hydrolyzed, and we have formation of 70S initiation complex. So there are three initiation factors that we saw here, that is initiation factor 1, 2 and 3 that is needed. Okay. Now let's see the elongation. Assembly has occurred. How the elongation takes place. Now when we say elongation, what do we mean? We are saying that how the elongation or the synthesis of polypeptide chain, how amino acids one after the other are added in the sequence. So that is what is elongation all about. So elongation also has 3 steps, and very interesting. Elongation is very interesting, where what happens is, briefly I will tell you the 3 steps. This is how the, at the end of initiation complex, this is what it looks like. You have the first initiator amino acid in the P site. You remember the middle one is the P site. So now the new tRNA, the new amino acid tRNA that has the amino acid for this second codon should enter, right? So that will enter at this A site, because this is the acceptor of tRNA. This is the amino acyl site. So this is where the new tRNA will enter. So that is the first thing that should happen. Second thing should happen, if this is the correct tRNA over here, there should be a peptide bond formation between this amino acid and the next amino acid, because that's how the chain will grow. Okay, so there will be the second step of peptide bond formation, and the third should be, once this is occurred, and the third step is the translocation. The chain formation has occurred. Now the ribosome need to move 3 nucleotides ahead, 1 codon ahead, right? So the 3 steps of elongations would be binding of amino acyl tRNA to the A site, peptide bond formation, that is the second step, and the third step is movement of ribosome to next codon. It should move 1 codon at a time. So let's see how it happens. Okay, so this is where we stop. So the first step is binding of the next amino acyl tRNA to the A site of ribosome. So what will happen is, this A site is now free, and that has the second codon. So the tRNA that has the anticodon for this particular codon should enter the A site. So let's say, for example, the second codon codes for amino acid 2. So this tRNA should enter the A site, and that happens with the help of elongation factor TU along with GTP. Okay, like we had initiation factors, in case of elongation we have elongation factors. Okay, so elongation factor TU stands for thermal unstable. So remember, along with EFTU in molecule of GTP, this next amino acyl tRNA will enter to A site. Now it is also possible that some mismatch tRNA might also come and try to sit over here. So that will not fit in, because the codon and anticodon match will not be there. There will be no hydrogen bonding, so it will be simply removed. So the correct tRNA would, with the correct anticodon will come with EFTU and GTP and come and sit on the A site. Okay, this much is clear. So once this amino acyl tRNA sits over here, what happens is the correct tRNA is there. So the next thing should happen is transfer of this amino acid over the next, and there should be a peptide bond formation. Okay. Now when this binding happens, there will be hydrolysis of GTP and this EFTU needs to be released, right? So this EFTU will be released and this EFTU can be actually recycled back to bring in a new tRNA, a new amino acyl tRNA. And that is recycled with the help of elongation factor TS. So now after this elongation factor is released, this is how it looks like. Right, both the tRNAs are there along with the amino acid. Now this amino acid needs to be shifted on this, right? The chain has to grow, so peptide formation should occur. So that is the second step, formation of peptide bond between these two amino acids. So there will be peptide bond formation between this F met to the next amino acid. And that is possible because the ribosome has the peptidyl transferase activity. It has the peptidyl transferase center. So it can shift or it can form the bond between the adjacent amino acid. So what happened is amino acid is shifted from P site to A site on the A site. So now there are two amino acids in this short peptide chain. So this results in both the amino acid being at the A site now. It is on the tRNA of A site. So on the P site, this tRNA is uncharged. It is free. Now this needs to be removed, this uncharged, because there we need this A site to be free so that new one can enter. So now there will be movement of ribosome to the next codon. That means we need to move the ribosome one codon ahead. And that movement is possible with the next elongation factor, that is elongation factor G with a molecule of GTP. So EFG with the GTP will enter and that causes hydrolysis of GTP, resulting into movement of this ribosome one codon ahead. You can see now it has moved this one codon. So what happened is the tRNA that was in the P site has now entered the E site, and the tRNA that was in A site has entered the P site. So now we have A site that is free once again, so that the new one can enter. From this E site, this uncharged tRNA can be removed easily because this is the exit site. The tRNA will be removed from the complex. So at the end now we have two amino acids. The next same thing will happen. New tRNA that contains the anticodon for this sequence would enter A site. There will be peptide bond formation. So this amino acid will be shifted over here. So you'll have amino acid 1, amino acid 2 and amino acid 3. Peptide bond formation will occur. Once peptide bond formation has occurred, ribosome will move one codon ahead and this uncharged tRNA will be released. Once again the cycle will be repeated. So elongation has three elongation factors that we saw. It will have elongation factor TU, that is thermo unstable, where it brings in the new amino acyl tRNA. Then we have EFTS, that is elongation factor thermostable, that will help to recycle this EFTU to bring in new tRNA. And then we have EFG along with GTP that helps to translocate, or that helps in the movement of ribosome. Alright. Now this cycle will be repeated one after the another, and you will have the long chain of amino acid or sequence of amino acid. And once it reaches a point where the reading frame is completely done and it reaches to a stop codon is when the termination will occur. So let's have a look at termination now. So in case of termination, termination is when it reaches to the stop codon, and as I said there are three stop codons, that is UAA, UAG and UGA. So when any of these three stop codon is reached is when the termination will occur, because there are no tRNA having anticodon for this three stop codon. So there will be no tRNA, protein synthesis will stop. Now in E. coli it is seen that we need specific release factors that can release the help in release of polypeptide chain from this assembly. So there are three release factors, that is RF1, RF2 and RF3. Release factor 1 can recognize UAA and UAG. Okay, stop codon UAA and UAG. RF2 can recognize UAA and UGA. Whereas RF3 does not specifically recognize any of the stop codon, but it helps or it stimulates in the termination event. It helps in the termination events. So based on what stop codon it is, RF3 along with 1 and 2 will help in the termination. I remember it this way, UAA is common. UAG, A is first alphabet, so it is the RF1. RF1 can recognize this, and then A is at the second position, so I remember it this way. RF2 can recognize UGA. So this is what happened, after the repeated round of elongation, you have the long sequence of amino acid. So in the end, when the A site will encounter this, there are no tRNA. So release factors would come and recognize this anticodon. Based on what anticodon it is, release factor will come and bind along with GTP. So now once this will come and bind to the A site, you remember in the elongation what happens when tRNA comes and bind to the A site, the next step will be peptide bond formation, where the amino acid sequence need to be shifted, right? But here there is a release factor, there is no amino acid. So peptide bond formation, for that the amino acid sequence will be cleaved from this P site, but there is nothing over here. So this amino acid sequence will be released. Right, so now this polypeptide is released from the P site, and the ribosome is again separated into 30S and 50S ribosomal subunits. So that's how the termination occurs. As I said, there are no tRNA that has the anticodon sequence of stop codons. So release factor would come based on what stop codon it is. The second step is, once there is something in the A site, the peptide bond formation. So this release factor, when it comes and binds, would result in release of this polypeptide chain. There is nothing here to bind, so it will be released. GTP is hydrolyzed. Ribosome is separated into 30 and 50S subunits, and release factors are removed. mRNA is free now. So that's how the termination of translation occurs. So that's all about translation in prokaryotes. I hope this video was helpful. Do subscribe to the channel for new video every week, and I will see you next time. Until then, keep learning.